So today I'd like to talk about the elephant in the room for most cures, and that is content. People always ask, how are they learning anything, or are they learning anything in your cures, right? You've had to drop out some sort of projects, you had to drop out all the different weekly lab where you're exposing them to all these different techniques, and if you do that, are they losing content? And so we need to figure out how to feed the elephant, right? We need to figure out assessment. This is really, really important. It's important for your students. It's important for you. It's also a good way to show other faculty and administrators that these types of labs are working. And so for my class, everybody does this a little bit different, but this is how I designed the synthetic biology cure lab. And it's really how I design most of the cure labs as far as assessment and grading is I like to have lots of oral presentations. Good way to figure out what they're doing. We have formal presentations where they stand up before the class, they have formal PowerPoints, and they present what they're doing. The first presentation in synthetic biology lab is, here's my project, here are the parts I'm going to put together, and here's what we might do. We also have informal type of meetings where they're talking to me. They're meeting with me, they're discussing their projects with me, and these are very informal, but it's a great way to see what's going on. And then, of course, we have other final presentations. I will make a quick comment also, is that students, as they meet with me, have to convince me they understand their project. If they do not understand their project or understand cloning, I do not let them in the lab. And so I have some students that start their project 3 weeks in. Other students will start 4 or 5 weeks in. But if they don't buckle down and they don't understand their project, it is not fair to them to let them start bumbling around in the lab. Once they go in and they understand the process and what's going on, they do so much better and their success rate is so much higher. And because of the benchmark grading system, it works really good with this as well. We also have lab notebooks and we have final written reports that students have to do if they completed their project. So we have lots of different ways to assess learning related to the lab. And so just as a general lab breakdown, a nuts and bolts breakdown, lab is 25% of my overall class grade. I divide that overall lab grade into 2 main sections, and points are contributing to one section or the other. The first section I call the lab project or notebook grade. This is a series of completed tasks related to the benchmarks that they have to hit. They have notebook they have to keep up. I grade the notebook early on, about midterm, and then I grade it again towards the end. And then if you complete your project, you have to write an analysis paper. If you do not complete your project, then you have to write a reflection of what you did and what problems you had related to it. I also have some grading that's not really tied to success in the lab. And so presentations. Presentations, you can be really bad in the lab, you can have a lot of problems in the lab, but you can give a good presentation. I also have a midterm sort of test your knowledge type of exam where I'm asking students to interpret results. So, here's a gel image, and what does this gel image mean? How would you clone part A and part B together? And I'm just trying to keep them all sort of honest, making sure as they're working in groups, they're still keeping up and understanding what's going on in the day-to-day lab. And then group meetings and assignments, a lot of times they have to bring outlines that they have to show me. These outlines are graded and they get some sort of points for showing up with these outlines, or they have to go look up something related to a BioBrick and bring it in towards the beginning of the lab. These are all assigned points, and these are kind of sort of a weekly quiz that's given for the first couple of weeks. I slack off once they start working on their project, but these are all things that I can grade and easily assess where they're at throughout the class. The other question I always get is about the lab portion of the grade, and that's where the benchmark comes in. And so some students may not complete all the benchmarks, and so they don't get all the points. And so what happens is you're working towards 50 points most days in the lab, or most semesters in the lab. And so, for example, successfully cloning 2 parts together. So that's putting part A and part B together. You have to verify it. You have to run a gel to show it's there. You've done all the parts to prove that you put part A and B together. You get 10 points. You create freezer stocks. I give you 5 points. And so freezer stocks is something new I've added. It's a great way for me to start freezing different parts back, creating components that students can build on in other semesters. I also have a degrees of difficulty system where many years ago I found that students were choosing projects based on how easy they were to succeed to get all their points. And I don't want them to do that. I want to encourage students to do more and do harder projects. And so if you choose a hard project, then I give you more points. I give you bonus points. So no matter where you finish up, those bonus points are going to be added to your overall grade. And so it's those benchmark points. So say you get 30 out of 50, plus your notebook grade out of 50 points, get added together, and that becomes your lab project grade. A lot of students will not get 100 on the notebook, and a lot of students will not complete the project. In my hands, most days, about 65% of the students completely finish this project. That's a lot. I'll also say that a lot of students still make A's and B's even if they don't complete, because they're doing all the assignments right, they're doing the presentation right, they have a great notebook, and they get really close. If you complete your project, then all you have to do is write an analysis paper showing how you completed it, documenting your success, and documenting your analysis, your experiments to test whether or not that promoter works, or whether or not green fluorescent protein works better in this system than red fluorescent protein, or whatever you designed, and that becomes your grade. And so the incentive here is that I'm not grading notebooks, and you may have missed something in your notebook, and you're more likely to miss points than you are to get points when I'm grading notebooks. And so really people are working towards not having to have their notebook graded. I'll also say that one of the things that I found is that if you're successful and you're able to get to that analysis paper, your notebook's pretty good, because you have to stay organized throughout the time. This is one of my favorite notebooks. It was from 2 years ago. And if you look in this far corner here, students write a summary of what they're about to do that day. They write a little analysis. Here's what we're going to do. I also have them draw what their gel image is going to look like. So they're going to run a gel today to verify their inserts. And so they wrote about what they're going to do. They also drew their image of the gel. I find this helps with interpretation of data downstream. And one of the things I really like is that students then notice that what they drew initially wasn't correct. And so they actually redo their gel, right? So now we have multiple iterations. They made a mistake, they caught their own mistake, and they fixed it. And they eventually ran a gel. At the same time, students are also designing their experiments that they're going to do for the analysis of this project. So they haven't done it yet, but they're already starting to think through and do their analysis. If you go a few more weeks into their lab notebook, here's the actual results from the experiment that they designed on this page. This was a really good project because students were looking at different promoter strengths, and they wanted to compare what does it mean to be a weak promoter versus a strong promoter. And so they took various weak and strong promoters and they added them to green fluorescent proteins. They also added some to red fluorescent proteins, and they designed these experiments. Which one of these promoters is really strong or weak, and do they work differently with green versus red fluorescent protein? We didn't use a plate reader in this situation. The students just didn't want to use it. And so they designed an experiment where they had others look at these projects and then look to see whether or not they're actually fluorescing. And so, yeah, there was some issues of not using a plate reader, but they just had a number system, and they devised this all on themselves. And then they ran a little bit of statistics, and they graphed their data. What I really like about this notebook is that these students were mid-B to high C students. They were not the 4.0 med school bound from the day one type of students. These students got connected, they got involved, and they really took ownership of this project. And so what I like about this is that you don't have to be able to memorize everything in the world and put it down on a piece of paper. This really is a great way to improve and encourage students. We also have other types of ways we assess. We do a major field achievement test in biology. We've moved to a different system this year, but however, when we were doing the MFAT test, molecular cell biology and genetics were the highest rated categories. Biochemistry ACS standardized exams, we see that molecular biology was one of the higher sections. One year we had an adjunct who did not even teach molecular biology in that section. That goes a long ways to get other faculty to see what you're doing is actually working. Jamie Sable will talk about some of our attitude tests that we are taking. We'll have you take a pre and post test. We're also doing a Cure Elements test that we do, that Aaron Dolan published, where are we hitting the different components related to a cure. Other types of ideas for assessment is in 2016, Jace Bradshaw was doing a Terminator project in the synthetic biology lab, whereas Alibel Gongola was working on a bioinformatics project in a different class looking for Terminators. And so we got to thinking, well, we can take Jace's idea from the BioBricks and can we make a better Terminator testing construct? And we did. And so we designed this project. We did some bioinformatics. We made our new Plasmid. We use a completely different cloning strategy than what BioBricks use. We can rapidly clone in Terminators and test what happens. And we base this on a process called Golden Gate Assembly. And there's other versions of this. And so I'll show you how these things work. But I think this is a good way of showing how we take what's in the lab and then things can be moved out into a research type of setting. And it was really cool. Jove came down, they wrote a script. They came down, they videoed us doing all the scripts, and now there's a video protocol. Jace's plasma now was submitted to AdGene, and it's been given to 20, 30 faculty all over the world have requested this plasma. So it's really been pretty cool to see what happens and how we move these things. And so assessment can be fun, right? At first, I used to think about assessment and I used to just hate it, right? Oh, how do you have to do this? But it really can be fun. It can be good, and it's definitely needed. Thank you.